All right, so we have just finished up talking about lipids, which is the second macromolecule that we are going to go over out of the four. Now we're going to move on to the third one, which is nucleic acids. Okay, so nucleic acids, this is where it gets a little bit different as far as the elements go. Remember the lipids that we had earlier? See here, the lipids and our carbs, they had the elements carbon, hydrogen, and oxygen, but they did not have nitrogen and phosphorus. I'm going to show you kind of where these guys come in a little bit later when talking about this image that we've got onto our right. So again, it's got the C, H, and O, but it does not have the N, the nitrogen, or the P, the phosphorus. Okay. As far as the structure of nucleic acids are made up of nucleotides. So we could also say that the monomer of nucleic acids, monomer right here, the monomer of nucleic acids is nucleotides. And then nucleotides, whenever we get all those together, the polymer of that is actually nucleic acid. So there's pretty much just one building block here. We've got our monomer of a nucleotide, and if we get a bunch of nucleotides attached together, then that's what creates nucleic acid. So the polymer is also the macromolecule itself. All right, but just kind of looking at this picture that we've got to the right here, this is a picture of a nucleotide. Okay, so a nucleotide is going to be super important when we start talking about DNA because our DNA is made up of a bunch of nucleotides, and this part of our DNA, or our nucleotide, excuse me, this base is actually what kind of makes us us. It gives us this little code that determines what our DNA does, or maybe some traits that we may possess and so forth. But we'll talk about that whenever we get into genetics. But I did want y'all to know that nucleic acid is closely related to DNA. If you hear nucleic acid, I want you to think DNA. And the monomer of that again is the nucleotide. So here's our picture of a nucleotide. I often tell my kids that, okay, here it looks like a house. All right. Here it looks like a little balloon. And then here, a lot of the times, it's going to look like a rectangle. Okay. Here it's a different image, but this is still a nitrogen base, but a lot of times it'll look like a rectangle, so I say it looks like a flagpole. Okay, so our house with a balloon, and it's also our house is attached to this flagpole right here, or this flag. To break apart this nucleotide, so our house is pentose sugar. Okay, sugar is a like it could be a carbohydrate of some sort. And then we've got a phosphate group right here. So our phosphate group is where that phosphorus comes into play. And our nitrogen base is where our nitrogen comes into play. So remember on our lipids and our carbs, we had that carbon, hydrogen, oxygen. We're adding this nitrogen and phosphorus because if we break apart that nucleotide, we see that those are those two components that happen to make it up. Okay? And then you could also see we've got some oxygen and some hydrogen here, and different things like that, to where that makes up all of those elements that we have right here. When thinking about the function, like I said, it makes up DNA and RNA. If you're like, oh, I know what that is, awesome. If you don't, that's okay. We're going to talk about this way more in depth whenever we get to genetics. But this is a chromosome, and if we were to unwind the chromosome and get in and zoom in as much as we could, right here it is showing us nucleotides attached to other nucleotides. Okay, like here you see our house with our balloon, and this would be our flagpole. Okay, so we just take a bunch of nucleotides, and whenever we get all those nucleotides together, that makes a nucleic acid. All right, because that would be the polymer. And the main thing is it makes up ATP. That's one thing, but the main thing here is that it stores genetic info. That's the whole purpose. Not the whole purpose, but a main purpose of nucleic acids is storing that genetic info. Some food examples. As I'd said earlier, that nucleic acids, what's cool about them is that they come from basically anything that was once living because they are in our DNA. Okay, so in a sense, like when you're eating a banana, you are eating the banana's DNA, what makes it that. So in that DNA, you're getting that nucleotide, which the polymer of that is nucleic acids. Then just any type of meat, plants, veggies, all of that good stuff is going to be a great source of nucleic acids. Next, we've got our final macromolecule is the proteins. And so right here, it says here the elements are made up of carbon, hydrogen, oxygen, and nitrogen. Okay, so think about which one do we not have in protein. So we know lipids, we had carbon, that's not a word, carbon, hydrogen, and oxygen. Okay, in carbs, we had carbon, hydrogen, and oxygen. In our previous one, nucleic acids, we had carbon, hydrogen, oxygen, nitrogen, and phosphorus, okay? But our proteins, we're going to take out that phosphorus. So we've got carbon, hydrogen, oxygen, and nitrogen, okay? That would be the elements that make those up. So the structure of proteins are super important. In everything that we do. But the structure of it is the amino acids can link together by a peptide bond in order to make a protein. Okay, so here in this picture, all these little balls right here, those are amino acids. All right, and if we were to zoom in even more, here it's got our amino acids. There's a ton of amino acids with different names. You don't need to necessarily know all of those. But in order for those to kind of stick together, it has to have this peptide bond, which kind of acts as the glue that's going to glue all these little balls together, which are going to make a ball link chain. And once you have that ball link chain, then you're going to have your protein. All right. And different proteins will code for different things and all of that good stuff. So the amount and order of amino acids determines the type of protein. So here it says we've got PHE, LEU, SER, CYS. So the order that these guys are in determines what type of protein is. If I were to just take out the CER and it was only these three guys, it would end up being a completely different protein. So how they are in order is super important. The amino acids, how they stay in order is very vital in order to make the correct proteins, which is going to get our body to do the correct jobs. Moving this over here so you can see it. Whoops, not that part. So here we've got, again, still talking about proteins. So proteins, the monomer of proteins is going to be amino acids. So remember those little balls that we had on there. The polymer is just going to be protein within itself. So again, we're not really taking another step up. We've got our building block of monomer, which is an amino acid, and then our polymer, which is like multiple Legos connected together, equals that protein. Some examples of protein are collagen and enzymes, and we're going to focus on enzymes a little bit later on in this presentation because they play a major role in making things that we need to happen in our body happen efficiently and fast. But those are all examples of proteins. The function of proteins is to provide structure, transport substances in and out of cells. We're going to talk about this more whenever we get into organelles, but basically proteins, there's these protein channels that allow certain things to come in and out of cells that normally can't. And then we've also got, last but not least, is to help fight off diseases. Again, some examples of proteins is collagen, hemoglobin, antibodies, amylase, and then some food references of that could be almonds or chicken, nuts and poultry. Any type of meat is going to have a great source of proteins. Now, again, I said we were going to focus a little bit on enzymes. Move it right there. We're going to focus a little on enzymes because enzymes are a type of protein that are going to help our molecules react with one another. So it says here that they're used by the cells to trigger and control chemical reactions. Basically, without enzymes, several of the reactions in cells would never happen, or they would happen too slow and it wouldn't be useful. An enzyme is mostly going to end in ASE. That's how you can kind of figure out. So like amylase, that would be an enzyme. Lactase would be an enzyme. Just some examples there. This image is a picture of an enzyme right here. This turquoise type thing. So it kind of looks like a Pac-Man. I don't know if y'all have ever played that before, but that was a big game back when I was a kid. So this little Pac-Man here is actually the enzyme. And what the enzyme does is it goes and let's say that this was lactase, okay? So then what's going to happen is maybe the body's got to break down lactase. This is going to be a substrate, which is just basically the thing that it's going to break down or speed up the process of. And what happens is they fit together like a key. The active site is kind of like where they all meet with one another. And then you're going to see this little process here. So our substrate fits into our little Pac-Man, okay? And then that's what happens, and the enzyme is going to speed up the process, which is called catalysis, whenever you're speeding up whatever process is going to happen. And later the enzyme is actually going to end up making a product, okay? So substrate is what we're starting with. The substrate is going to be kind of like the key to the enzyme. The enzyme then is going to connect together at this active site. It's going to speed up whatever reaction it's needing to speed up. And then it's also going to not reproduce, but produce two new products. So two new things. What's coming in is going to be different than what's coming out. And it makes that whole reaction for whatever type of substrate is coming at it, it makes it to where it goes a lot faster. So we definitely need enzymes. Real quick, just to talk a little bit about what they are, the specific parts of it, because being able to label and know what enzymes are and just kind of the process of that is important. So it says enzymes bind to or break molecules called substrates. So they're going to bind or break. And these substrates are the reactants that are catalyzed by the enzyme. So basically, those are going to be the things that are going to be sped up. The active site is going to be where that substrate and enzyme have a specific shape. It's going to kind of be like a puzzle. Okay, see how the substrate has the jags in order to fit into that enzyme? So that's the whole point of the active site, is to basically be that puzzle piece to where they fit to one another. And the site on the enzyme where the substrates bind, which is that puzzle piece, is called the active site. Okay, so just some examples of enzymes. So lactase is one. I use that as an example. It breaks down lactose into glucose and galactose. You don't need to know all these. This is just some examples. So let's say that I said, all right, this substrate is lactose. It's going to come into lactase, this enzyme. It's going to be sped up, and then it's going to break into the two parts, okay, of glucose and galactose. And then these are also just some examples, sucrase and amylase. But notice that they all pretty much end in ASE. All right, and then just kind of to check ourselves, because you'll probably see images like this where it's basically showing you the process of an enzyme. So we've got our enzyme here, okay, so that would be label C. This is showing the part of the enzyme. I want you to take a guess at, it's the part of the enzyme where it's going to basically kind of be that binding site for the substrate and the enzyme. So I want you to think about what that could be. Then here we've got something that's going to fly in. Let's say maybe it's lactose, and it's needing to bind to this lactase in order to speed up that reaction. All right, then it is being sped up, and now the enzyme is going to break that lactose, and now it creates the two different other molecules that was needed. And these two different molecules are called the product, okay? And then we've got our enzyme, and it's ready to do the same job all over again. So enzymes can be reused. Last thing, I just want to kind of check for some understanding. So this is a great little slide to kind of jot down. This basically shows you the different elements, all right, and the cellular functions. And what I want you to do is we're going to take this last little time I want you to tell me, okay, what is A, what is B, what is C, and what is D. Y'all let me know if y'all have any questions. Again, I am here for y'all. Just make sure that we figure out what A, B, C, and D are, what macromolecules they are. Just to review, we have lipids, we have proteins, we have nucleic acids, and we have carbohydrates.